Okay, so thank you everyone for joining the Paleoclimate Advances Webinar Series. And it has been a while since the last webinar, but we have been working and having discussions behind the scene on how to better organize the webinar series. And I'm happy to announce that today, Sophia Makarowicz has joined the steering committee to help us organize the webinar series in the future. And Sophia is a colleague of mine. She's a project scientist at NCAR, and she will help us host today's webinars. Sophia?
Great, thank you. So by joining today's webinar, you agree to follow our code of conduct. So this basically means that during the webinar, please offer constructive feedback, consider new ideas, show appreciation, encourage innovation, acknowledge teamwork, and share the air. So the webinar is structured to have time for 2 20-minute talks with 5 additional minutes for Q&A. Towards the end of your talk, when there's 2 minutes remaining, I'll give a reminder just to make sure we stay on time. And then at the end, we should have 10 additional minutes for discussion. Our co-host Sijun Sun is going to help moderate the Q&A today. And then also, I just want to remind people that we always welcome speaker nominations. So please, if you have a speaker in mind that would be great for this webinar series, please go to the link shown here and nominate speakers. So it's my great pleasure to introduce today's 2 speakers, Dr. Spencer Jones and Andreas Schmittner. Dr. Spencer Jones is an assistant professor at Texas A&M University in the Department of Oceanography. Spencer is a physical oceanographer whose research focuses on ocean dynamics and explores how the ocean transports heat and other tracers around the globe. He uses a combination of theory, ocean models, and scalable analysis tools in Python. Much of Spencer's work investigates the strength, geometry, and variability of the meridional overturning circulation. And Dr. Andreas Schmittner is a professor at Oregon State University in the College of Earth, Ocean, and Atmospheric Sciences. Andreas's work, there we go, focuses on oceans, their physics, biology, chemistry, and variability on decadal to millennial timescales. He tries to understand the causes for today's global deep overturning circulation, how and why it has changed in the past, how it may change in the future due to natural or human influences, how it impacts climate, ecosystems, and biogeochemical cycles, including isotopes. To address these questions, Andreas develops, improves, analyzes, and uses numerical models. So without further ado, let's start the webinar with Spencer. I will stop screen sharing. And please, Spencer, take it away.
You are muted.
Sorry. Okay. Thank you very much for inviting me. I'm gonna talk about some work that I've been doing, and the kind of broad theme of the work is assessing the large parameter space of possible LGM ocean circulations. And this is kind of a combination of 2 papers, one that I wrote with Ryan Abernathy and one that was co-authored by me and Frankie Pavia and Sophie Hines. And yeah, so this is the kind of work that I've been talking about. But before all of that, I want to kind of set the scene. And I think, the kind of general understanding that we currently have is that during the modern era, right, we have North Atlantic deep water that's formed in the North Atlantic, right? If this is depth and this is latitude, this is the north. And so we have lots of North Atlantic deep water coming into the deep ocean. And then at the LGM, it seems like, at least based on Delta C13 observations, there was less North Atlantic deep water, and the peak of that North Atlantic deep water seems to have been a bit shallower than it was now, at least according to these delta C13 observations. And we kind of want to have an explanation for that. And the explanation that we have right now is that in the modern ocean, right, we have North Atlantic deep water that sinks all the way to kind of 3,000 meters depth, maybe like 2,500, something like that. And that is allowed because we have an Antarctic bottom water cell, an abyssal cell that is a bit less dense. And in the last glacial maximum type ocean, right, perhaps there was more sea ice, perhaps there was more buoyancy loss in the far south, and that led to denser Antarctic bottom water. And it meant that the North Atlantic deep water ended up above the Antarctic bottom water. And so you end up with this kind of shoaled amok cell or upper cell of the circulation. So this is kind of a very nice explanation of what we saw in the previous slide, that shoaled North Atlantic deep water was caused by, and this is not what I mean, it was caused by the upper cell shoaling and allowing the deep ocean to fill with Antarctic bottom water. And this is also very nice because it sort of gives us this, there's this explanation for how CO2 levels might have changed. One part of that might be that there was more carbon storage in the deep ocean. And so when you look at, this is an idealized model that was run by Alice Marzocchi and Malte Janssen. And I really like this study. It sort of shows that if you have a more isolated abyssal cell, then you often end up storing more carbon in the abyssal ocean. And that means that you end up with less carbon in the atmosphere. So right, the abyssal cell may have controlled atmospheric CO2 levels, and the speed of circulation also matters. I don't know whether this is what Andreas is going to talk about, but I thought I'd mention it, that right, the speed of the circulation also seems to matter for carbon storage. So this is kind of a very nice explanation of what's going on. And I kind of want to frame this as being like the hypothesis, like, right, our hypothesis is that North Atlantic deep water shoaled because of a change in the large scale circulation. But there are actually quite a lot of sources of uncertainty here. And I'm kind of, the rest of the talk, this is kind of an outline of the rest of the talk, is about thinking about what kind of uncertainties there might be and what other explanations might be possible for the observations that we see. And so, right, one of the sources of uncertainty is not all paleotracers agree about what kind of fraction of North Atlantic deep water there was in the deep ocean. Not all models agree that the upper cell of the AMOC shoaled. And then other factors that could be important are, right, models disagree about water mass concentration in the deep ocean, even in the modern era. So it's a huge challenge to sort of model even the modern deep ocean circulation. And it's that makes it kind of even more of a challenge to then sort of take that and think about the past. And it seems like one reason why this is a problem, this is difficult, is isopycnal and diapycnal mixing also influence deep ocean tracer distributions, and they're kind of poorly constrained even in the modern era. So how are we supposed to know about what they were like 20,000 years ago? And then I kind of, this is kind of giving away what my final conclusion is, but I think more different GCMs and idealized models are needed to kind of understand the breadth of the parameter space. So first, right, I'm going to talk a little bit about the different tracers, and the tracers that I'm really familiar with are two tracers, the delta C13, which we talked about before. And so delta C13 is influenced by multiple factors, including biology, but people are trying to correct for the effects of biology using cadmium. The data is a bit limited, and so there's some uncertainty in understanding what was the distribution of delta C13 when you've corrected for the biology. That's already a little bit difficult. And then there's this other tracer, which is I think becoming more people are more interested these days, which is epsilon neodymium. So right, neodymium is a tracer in the ocean, and epsilon neodymium describes the kind of fractionation of the isotopes. And the ocean floor, the kind of complexity for that is that the ocean floor may be a source and sink of epsilon neodymium, and so that therefore it might not be a completely conservative tracer. And so this is a diagram that Sophie Hines made, which kind of shows, right, we are expecting there to be two kind of end members. One end member is in the North Atlantic, and it has sort of high delta C13, and it also has low, which means kind of more negative neodymium isotopes. And then near Antarctica, you're expecting to have low delta C13 and more positive neodymium isotopes. So we have these two different tracers. And it turns out they actually agree pretty well in the modern ocean. So you can see, right, this is the Atlantic. This is delta C13 SC, which means you corrected for the biology. And you can see there's kind of like a blob of North Atlantic deep water, and that's in agreement with the epsilon neodymium that people are measuring. Basically, this area seems to have more North Atlantic deep water, and then the southern end has less North Atlantic deep water, and you see that there's kind of Antarctic bottom water sort of coming in the abyss, but it only extends to around the equator. When you look at the LGM, the delta C13 SC is what I showed before, right? It's kind of suggesting that you have shoaling of the North Atlantic deep water, and more of this southern source water is coming in. Now you'll notice, right, the sign has actually, the direction of the relationship has changed. And so in the LGM, we actually have the opposite sign direction of the relationship between delta C13 SC and whether you have more or less North Atlantic deep water. But I mean, the argument stands, right? This seems to be consistent with the shoaling of North Atlantic deep water. But then when you look at neodymium, it seems like there's more northern sourced water in the deep ocean. And so this water seems to be coming from the surface of the North Atlantic based on the neodymium, epsilon neodymium concentration. And so, you know, these two things don't really line up. And that's one reason why we might not 100% trust the kind of current explanation, is that it doesn't actually fit necessarily all the data without doing a bit more work. And then, right, not all climate models agree about the LGM circulation. So these are just three that I picked out from the array of possible simulations that you could pick. And some of them have no change in the depth of the circulation between the pre-industrial period and the LGM. Some of them actually have deepening. So the yellow in these plots is the pre-industrial, and the black is the last glacial maximum. And so you can see there's a little bit of deepening in this GISS model of the circulation during the LGM period. And then this is a case with CCSM4, where the yellow is the pre-industrial and the black is the LGM. And so you have shoaling of the circulation during the LGM. So some models have shallower AMOC, and some models have deeper AMOC at the LGM. And even, right, in the modern ocean, it's actually really difficult to say, oh, we're seeing this much North Atlantic deep water in the deep ocean. If you compare these two models, which have really quite similar, so right, remember the yellow is the pre-industrial stream function. They have this really quite different, this really quite similar circulation. But then when you look at the concentration of North Atlantic deep water in the deep ocean and try to estimate that, you will probably find quite different estimates of concentration of North Atlantic deep water in the deep ocean. And that's really because tracer distributions are controlled by multiple factors and not simply by the shape of the circulation. And so what could those factors be? Well, What I'm going to talk about now is the 3D distribution of mixing is important and may have been different at the LGM, and the mixing can influence how much North Atlantic deep water you find in the deep ocean and how much Antarctic bottom water you find down there. And then, right, there are some other factors like the surface boundary conditions may have been different and likely were at the LGM. And yeah, there's going to be various things like dust and the effects of sea ice on air sea gas exchange. And I am not a chemist, so probably some of you know more about this than me. But yeah, lots of different factors could be important. And then also biological factors may have been different at the LGM. So I'm not going to talk extensively about vertical diffusion because that's not what my work is about, but it seems like vertical diffusion has a huge impact on traces. So right, these black arrows are supposed to be the large scale circulation, and then on top of that you have traces being moved by vertical mixing, which we now know since kind of the early 2010s, vertical mixing is really increased towards the bottom of the ocean, and so we're kind of expecting that distribution, which is actually quite a complex distribution that depends on the topography, is going to influence the tracer distributions. And then there's this complexity that the sea level changed at the LGM, and so that may have caused vertical diffusivity in the deep ocean to be larger because you might have more dissipation of the tide in the deep ocean. But what I'm going to focus on right now is just isopycnal mixing, which is mixing along isopycnals that's caused by mesoscale eddies, and it moves traces along the isopycnals and down gradient, right? And so that also could influence the strength of the amount of North Atlantic deep water in the deep ocean. So I looked at isopycnal mixing in an idealized model, and what we did was we took this kind of idealized setup. We used some idealized passive traces, which are roughly equivalent to North Atlantic deep water and Antarctic bottom water, and we compared the modern circulation with a shoaled Atlantic meridional overturning circulation with multiple mixing scenarios. And so we have this kind of idealized setup. We have idealized surface forcing for wind, temperature, and freshwater flux. And then we also have an idealized vertical diffusivity. And we end up with these two, we're kind of comparing these two circulations. We have this circulation, which is more like the modern ocean, right? We have deep water formation both in the North Atlantic and in the Southern Ocean. And our North Atlantic deep water goes all the way to 4,000 meters depth. And occupies the deep ocean in the North Atlantic, but then it kind of upwells to the surface in the south. Whereas we have this other case, which is the shoaled upper cell. We decided that we weren't going to call it the LGM circulation because we don't want to we're not 100% sure what the LGM circulation was like, right? But this is the case with an upper cell that's shoaled, right? And so our North Atlantic deep water is only going to a kind of 1,500 meters depth, and we have Antarctic bottom water filling. The Antarctic bottom water cell is filling the whole of the Atlantic. So, right, this just because the cell fills the whole of the Atlantic, it doesn't mean that the Antarctic bottom water does. And that's what I'm going to show on the next slide. So this is the fraction of North Atlantic deep water that we find in each of the two cases. So here, this is the modern ocean, and we find that because the upper cell, the AMOC cell, is going all the way to the deep ocean, our deep ocean is in the North Atlantic is just full of North Atlantic deep water. So when it's 1, that means it's just 100% North Atlantic deep water. And then when we have a shoaled upper cell, that concentration goes down. We end up with less North Atlantic deep water in the deep ocean. But it doesn't fall to 0, right? So there is still quite a lot of exchange between the two cells. And that exchange, I'm not going to show too much about that, but it seems like that exchange is probably being mediated by diapycnal mixing. And like we said, diapycnal mixing, it was probably different in the past. And so the size of the transfer of tracer between the two cells probably did depend on vertical mixing and kind of the details of topography, which makes this whole question kind of difficult. And then, the other thing that we did find, where we changed the isopycnal mixing strength in our simulation, and we found that it had a large impact on deep ocean tracer distributions. So this is the change in North Atlantic deep water concentration for the high isopycnal mixing case minus the low isopycnal mixing case. And so you can see in the modern ocean and in the shoulder Purcell case, when we increase the isopycnal mixing, that reduces the amount of northern source water in the deep ocean. So the amount of North Atlantic deep water goes down, which is what the blue means. The amount of North Atlantic deep water is going down, and the amount of Antarctic bottom water is going up when we increase the amount of isopycnal mixing. And that seems to be because we have more exchange. That increasing the isopycnal mixing increases the amount of exchange between the surface of the Southern Ocean and the deep ocean. So we know that, isopycnal mixing may have been different at the LGM. Changing winds often have an impact on the strength of eddies. And sorry, more typos, but we found that even if isopycnal mixing only changed in the top 2,000 meters, it would impact deep ocean tracer distribution. So this is a case where we only change isopycnal mixing in the top 2,000 meters. And we did that because we think that the effects of winds on eddies is probably confined to the top 2,000 meters, but it still has this impact on tracer distributions in the deep ocean. So it seems like, yes, isopycnal mixing might play a role here. Okay, I'm gonna wrap up. Some future directions. If we wanted to explore a broad range of parameters, analytical models can be really helpful for that. And I just wanted to highlight two.
Sorry.
Yeah, I just wanted to highlight two. This is one from Thompson et al. 2016. So there's kind of multiple layers, 4 layers. And there's the model has physics, right? And so it knows about the slope of the isopycnals in the Southern Ocean. And you're able to kind of model how the transport of North Atlantic deep water impacts the thickness of these layers. Another one I wanted to highlight was the one that we worked on in Jones and Abernathy 2021, which is a less of a dynamical model and more of an advection diffusion model, where we looked at the effect of increasing isopycnal mixing on diffusion, and we were able to replicate that in a really idealized model. So when we increased the amount of isopycnal mixing, we had more movement of North Atlantic deep water towards the surface of the Southern Ocean, even in our analytical model. So these kind of models are really helpful because they're kind of cheap and you're able to explore a lot more of parameter space. And I hope that I've kind of illustrated to you, right, there are so many different factors that impact tracer distributions that it can be really useful to take a broad perspective. But I don't think that's the only thing we should be doing. And running more models, particularly kind of long runs with multiple paleo-relevant tracers, will also help us to understand the breadth of the parameter space. Analytical models can only do so much. You can see the whole parameter space, but you probably can't be as certain about them as you would if you were able to run a full 3D model. And I'm also interested in looking at how to create transport matrices from model output and sort of run tracers offline. And I think that's another way to explore the parameter space, and particularly to explore how boundary conditions on these tracers, which don't influence the circulation, could have changed. So yeah, that's another thing I'm interested in. So I'm kind of just repeating this slide, and that's because I kind of told you the conclusions before I started. Right, we have this hypothesis that North Atlantic deep water shoaled because of a change in the large scale circulation. And I don't want to be too critical of that hypothesis. It's like a really elegant hypothesis, and it explains some of the results very well. But it doesn't explain everything. And so hopefully I've shown you, right, there are some things that are not explained. The different paleo tracers don't necessarily agree. And then also there are other factors that are much harder to pin down. So isopycnal and diapycnal mixing are a little bit harder to pin down because they have this kind of 3D distribution in the ocean, but that could influence deep ocean tracer distributions and could be, we don't know how big those factors are compared with the changes in the large scale circulation. So yeah, I'm really looking forward to continuing working in this space. And yeah, if you want to reach out to me, please, do. And yeah, these are the 2 papers that this talk is based on. Thanks very much.
Thank you. Now we're going to go into about 5 minutes of questions. And CJ, will you help us moderate the Q&A?
Yeah. Thank you, Spencer, for this awesome talk. And for the audience, if you have any questions, you can feel free to put it in chat or raise your hand and ask your questions. Because people probably need a few seconds to frame their questions or type their questions. Well, I can start by asking a question. So I was wondering, how does the isopycnal mixing would affect large-scale circulation? Is there an impact at all?
Okay, so this is a really good question. And in fact, we kind of removed that factor when we did these simulations. We just changed the effect of isopycnal mixing on the tracers. We didn't change the effect of isopycnal mixing on the circulation. And I'm actually currently doing a study where we allow the circulation to vary. But yeah, there's been a couple of other papers that also have looked into this in more realistic models. And the answer is yes, the circulation does change a little bit when you change the isopycnal mixing. But it seems to me that the main impact on the tracers is at least, it depends on exactly where you are in the parameter range of isopycnal mixing. It can have a huge impact if you change it a lot. But probably you have to change it really a quite unrealistic amount to have a large impact on the circulation. But yes, it does impact the circulation and it impacts the tracers. Probably the tracers a bit more than the overall circulation.
I see. Thank you.
Yeah.
Do we have any other questions? Oh, we have a hand up. Keith. Please feel free to.
Hi, Spencer. Thanks for that great talk. Perhaps you said this and I missed it, but what resolution were you able to run your idealized experiments at?
So we were still running at 1 degree resolution, and in some ways, the reason we chose, you can run idealized models at an even higher resolution, but we were exploring this question of what if you change the isopycnal mixing independently? And so you really need to be in a regime where the eddies are not resolved in order to look at that. So yeah, we just start with 1 degree.
Sure. Thanks. That makes sense.
Great. If people have any other questions, please. Oh, John.
I have a follow-up question about the resolution. So if we run higher resolution, does that mean the isopycnal mixing is better? Or
Yes, so I think
required?
that I perhaps skimmed over is the isopycnal mixing is something that is a parameter that is put in, right? I mean, the same with vertical mixing, right? If you could resolve everything in the ocean, right, you wouldn't have to have isopycnal mixing. It's representing processes that happen at the subgrid scale. And so if you have a higher resolution model, yes, certainly, you can you will get the isopycnal mixing to be more realistic. At the same time, even with the idealized model, we would have only been able to run it a few times with a higher resolution. We probably, and with a kind of fully coupled climate model, running at kind of 1/10 the degree resolution is something that is very hard to do, right? And so yes, it would be more realistic, but I think it's a huge challenge to run for long enough to get your traces to be equilibrated. That's a reason why people don't do it.
A quick follow-up. How high is the resolution we need?
So I think if you go to 1/10 of a degree, you will resolve the eddies in across most latitudes. I mean, not at very high latitudes. And this is really, I mean, this is kind of the next step when people are trying to model the modern ocean circulation, right? They're trying to go to 1/10 of a degree so that they can really like resolve this stuff, the mesoscale eddies. That's probably enough to really to solve a lot of these problems, I would say. It's still like there are going to be issues, and actually this is something I'm interested in, right? When you get to really where the Antarctic bottom water is formed, you need to resolve, because you're not actually resolving the eddies very well in the far south, right? How is salt getting into the Weddell Sea or wherever your deep water is being formed? This is still an issue, and but ultimately, like, yeah, I don't think that's going to be resolved anytime soon. Is the truth.
Thank you.
Yeah.
Okay, that's about 5 minutes. So if you have additional questions, save it for the discussion at the end. And now let's move on to Andreas's talk. Great. All right. All right, take it away. Thank you.
Okay, can you hear me now? Okay. Okay. Well, thanks for inviting me to this seminar series, and also for thanks, Spencer, for a great talk and introducing the topic of AMOC and glacial AMOC. We've been working on this for quite some time now, and I added Juan Muglia here as a co-author because I'm going to show lots of his results. But there's also other people that we've collaborated with, so a lot of people actually have contributed to some parts of this work. And to kind of set the scene here, what I'm trying to focus on is the early parts of the last deglaciation. So we're going from the last glacial maximum to Heinrich Stadial 1. So Heinrich Stadial 1 is approximately between 19,000 and 15,000 years before the present. And from the paleoclimate data, we know that during that time, the global temperature started to increase, whereas with the southern hemisphere leading and the northern hemisphere like lagging in the temperature rise. And at the same time, CO2 started to increase during that Heinrich Stadial 1, and the dust fluxes, as recorded here in Antarctic ice cores, decreased over that time period. And there's this evidence from Protactinium thorium records and other paleoclimate data from the ocean interior that the AMOC was reduced. This is the famous McManus et al. 2004 paper. I've just looked it up. It has been cited more than 2,000 times. And they have interpreted this this level here of the Protactinium thorium is around the production level, and they have therefore interpreted this as a collapsed AMOC. But we will see other evidence for the state of the AMOC during the Heinrich Stadial 1. And of course, over that Heinrich Stadial also, the sea level started rising. So lots of things were going on there in terms of climate and ocean changes, and trying to understand really how the AMOC changed and how that all affected the carbon cycle is something that is still not very well understood. And so for this presentation, I thought I'd go back a little bit in time to the year 2015, when I published with Dave Lunt this paper in the climate of the past. And what I did then, so that's 8 years ago or something like that, is I just did hosing experiments with a model where we had included C13 in the model. And so with different hosing amounts, you could get different AMOC perturbations, cases where the AMOC just reduced a little bit, and then cases in which it collapsed completely. And in the collapsed case, because we have a fully coupled carbon cycle in the model, what we saw then, atmospheric CO2 response increasing. And we also have carbon isotopes, so we can simulate interactively atmospheric C13 of atmospheric CO2. And C13 of atmospheric CO2 decreased in these simulations. And the black line and the black symbols are the paleoclimate data from ice cores. So kind of similar to the ice core data, but the caveat in that study was we're starting from pre-industrial conditions. So the model CO2 is actually at 280 ppm, whereas the observations, the CO2 is at 190 or something like that. So that was an issue. But if we looked at the interior C13 distributions, and in particular, this figure shows the changes, Heinrich stadial 1 minus LGM. And the top panels in the left is the Atlantic, and on the right, the Indian Pacific. So I've tried to collect some data, and I've gotten 26 benthic C13 records together for this paper. And if you look at that pattern, you could see a decrease in C13 in the Heinrich stadial in this upper intermediate ocean, North Atlantic. But if you look in the model, in the collapsed case, which is shown in the panels below here, there was a much larger decrease in the model. I mean, whereas the pattern matched kind of the decrease there with the decrease in the data, but in the model, the amplitude of the C13 reduction was much, much larger than in the data. But there was a high correlation, spatial correlation. So that kind of indicated to me that, yes, there was an AMOC change, but maybe not as strong, or maybe it was because we used these pre-industrial initial conditions that were wrong. Meanwhile, we've done some more work to explore these different questions, and we've added some data. And this is a paper that Juan Muglia led that just appeared in Nature Scientific Data. And so now we have 287 sediment cores, high-resolution sediment cores from all over the world's ocean. This is an international effort of the PAWS working group, OC3, Ocean Circulation and Carbon Cycling. And we have been working many, many years on this, a large group, as I mentioned, but the database is now available. And if you're interested here's the link to the database. And perhaps, I don't know if somebody can put it in the chat, the link, and I can do that later too, but so there's a lot more data, from 26 to 280, so a factor of 10 more data now. And then we did analyze some of the data, and here's a paper led by Janne Repschläger from the OC3 group as well, where she looked at the Atlantic subset of the data, and what she concluded there, or we concluded, is that there was still active North Atlantic deep water formation during Heinrich stadial 1. And to me, the most convincing evidence is the C13 distribution in the Heinrich stadial 1. There's still this high C13 indicated in the orange and red colors in the North Atlantic that we do not expect there if there wouldn't have been deep water formation. So, maybe during Heinrich stadial 1, the AMOC was not collapsed, or at least not as much as previously thought, reduced. But really putting numbers on it is very, very difficult. We've also made some progress in modeling more realistic initial LGM conditions. And this is, again, a paper by Juan here in 2021, where he produced different experiments with different AMOC strengths on the horizontal and AMOC depths in the vertical. And then we looked at the match with the LGM data. And this shows the root mean squared error. So lower values in orange are better, and blue values are higher errors, so are less fitting the data, are worse, are not as good fitting the data. And what we could see then in this AMOC depth, AMOC strength space is that the data really constrain the AMOC depth very well. So these deep AMOC simulations can be excluded, but the data do not constrain very well the AMOC strength. So, you know, if you have a shallow AMOC, a circulation with 9 Sverdrup is about as similar as in agreement with the data as a circulation with 17 Sverdrup. So the AMOC strength remains difficult to constrain. And here, so for the new simulations that I'm going to present today from Juan, he used this LGM circulation that has a similar strength of the AMOC as the pre-industrial run, but it is shallower. And an additional forcing that we need to fit the C13 data, and also nitrogen isotopes, I should say, we have nitrogen isotopes in the model, and they provide additional constraints, particularly on iron fertilization. I'm not going to show these data, but it's published. So so the nitrogen isotopes suggest, and the carbon isotopes as well, that we need iron fertilization, especially in the Southern Ocean. We need to put more iron in. And if we do that, the phytoplankton concentrations increase here, and we get more, we get better fits with the C13 and the nitrogen isotopes. So those are the basic features of this LGM initial conditions: shallower AMOC and increased iron fertilization. So that's our starting points for these deglacial simulations of the Heinrich Stadial 1. And I just thought I'm going to go back and look at the 2015 paper again and compare the new initial conditions with that one. So if we look at, this is a figure from Lund and Schmittner 2015, and the LGM data, this shows just profile data from the Brazil margin as an example here. That from David Lund. So the LGM is the black curve. These are the data, the dashed line. And then in the 2015 paper, since we used pre-industrial initial conditions, the model, you can see, has quite a different C13 distribution. So particularly in the deep ocean, well, first of all, it is higher everywhere, but in the deep ocean, it has a weaker vertical gradient than the data. And if we now look at on the right-hand side, the new simulations from Juan, the pink is, again, the Brazil basin data from the sediment cores. There's several simulations that fit these data quite well, and those are featuring intermediate and shallow AMOC. So there's definitely an improvement in the initial conditions from that previous paper, but with the caveat that we still cannot constrain the strength of the AMOC very well at this point with these data. So now what Juan has done is he has used also more realistic deglacial forcing. So he starts with the LGM. So now time goes from right to left. So we're starting at the LGM and then moving towards the left in time towards the Holocene. So he starts at the LGM simulation. The AMOC strength is around 17 Sverdrup, and then with varying degrees of freshwater forcing, the spatial distribution is realistic, but modified amplitude gives us different AMOC strengths. So the light blue, the cyan color here, is a collapsed AMOC. And then the green, the gray, and the orange colors are reduced AMOCs for different periods. And then the red is just a control run, and the blue is a control run where the AMOG is almost not changed. The blue one is a one where the circulation is not changed, but the dust input has been removed. And you can see that effect here in the upper right panel that shows Southern Ocean phytoplankton. So that removal of dust forcing leads to a decrease in productivity, a decrease in phytoplankton biomass. So we're exploring the effect of that in addition to the AMOG variations. And then there's combinations. So for example, the orange one is a weak AMOG only. And then the gray simulation is a weak AMOG plus removed dust forcing. And you can see here how the Southern Ocean productivity goes down in there as well. And I should say that even if the AMOG is weakened or collapsed, so that circulation changes also affects productivity in the Southern Ocean. So not only dust, but also circulation affects productivity in the Southern Ocean. Okay, so now if we look in these lower panels, in the lower right is atmospheric CO2. Now we're starting from more realistic values, about 190 ppm in the LGM. And the CO2 data, the symbols here show they go up by about 30 ppm in the Heinrich Stadial 1. And as you can see, none of the models reproduces that increase, the magnitude of that increase. However, the one with the dust forcing, the dark blue curves, matches the data best of all the simulations. And that indicates there may have been a role of dust forcing in this initial CO2 increase in the early deglaciation. Also note that the collapsed case, so the cyan line here, leads to a decrease in atmospheric CO2. This is different from the Schmittner and Lund paper in 2015, where we started from pre-industrial conditions and we get an increase in CO2. So that seems to be very sensitive with respect to the initial conditions. Now on the right lower panel, there's C13 of atmospheric CO2 from the ice core data. And here again, the blue line, so the dust forcing, matches the observed decrease in C13 better than all of the other models. Okay, so those were the ice core data. Now, the interior ocean, the benthic C13 data, kind of a summary plot is shown here that shows the root mean squared error over the evolution over 1,000-year intervals from the last glacial maximum on the right to Heinrich Stadial on the left. And for these different simulations, and I've indicated here in the dashed lines, this is what we expect the error to be for the pre-industrial combined model and data error, about 0.35 to 0.4 per mil. So if it is in that range, we would say the models are as consistent with the data as the pre-industrial model simulation. As you can see, none of the models are really, except here, this one that dips down here for this one time period. But even the LGM simulations, they have larger errors than the pre-industrial. So there's still something not quite as good as for the pre-industrial. But then if we move into Heinrich Stadial 1, you can see all of the errors, all of the models increase the errors, in particularly the collapsed state. So the collapsed state increases very much, has errors about twice as much as we would expect for the pre-industrial. So clearly we can exclude that scenario. But still the other models, even the one, the red one is the control run, so where the circulation does not change from LGM, even that increases. So that indicates that a constant circulation is not a good fit either. So, but the ones that fit the data best are the weak circulations here in gray and orange and green. But also the one with the dust forcing fits the data okay, but not as good as the one where it is the dust forcing and and a reduced AMOC. So that is the best fitting model, but still it is not fitting as good as the pre-industrial expected. So I think what we can conclude from this is that we can exclude a collapsed AMOC, but we still have not found really a simulation that matches the data as well as we want to. If you look at the spatial distribution, this is just showing the Atlantic data, and the sediment data is shown in the top left panel here. And what is shown here is the differences. So it's Heinrichstadial minus LGM C13. And there you can see in the data, again, similar as what we saw in the Schmittner Lund paper in 2015, a decrease in C13 in the upper ocean mid-depth North Atlantic. But now there's also,
sorry.
Yeah, thank you. In the deeper parts, there's some increases seen. And here, if you look at all the models, none of them really matches that pattern very well. The collapsed one obviously has much larger decreases, so is not consistent with the data at all. The control run does not have much change, is not consistent with the data either. And the best fitting models are the ones that have a weaker circulation and have dust included. So this one here, but even in this one, you can see the decrease in the model is deeper than in the observations. So there's still work to do, I think, but at least we can exclude some scenarios. Okay, so what we conclude from these is, I think, what is pretty certain is that during Heinrichstadial 1, the AMOC was not collapsed. That is a pretty solid conclusion. The collapsed AMOC would imply much lower C13 in the deep North Atlantic, and those are just not observed. But the AMOC may have been reduced somewhat, but still we cannot put in any numbers or quantitative constraints on the AMOC strength during Heinrichstadial 1. Yeah, so none of the simulations fits the Heinrichstadial 1 data and the ice core data. So if you remember the ice core data, the ice core data were fit best with the simulation that it had only dust forcing and no AMOC changes, whereas the interior ocean C13 data fit best a model that had dust and a weaker AMOC. So there's not quite consistency between these two different data sets. Something I think that we can maybe cautiously conclude from here is that a reduction in dust forcing may explain the initial CO2 increase, but that still remains, I guess, more like an hypothesis than a real conclusion. Okay, I think that's it. Thank you so much for your attention.
Thank you, Andreas. This is an awesome talk. And yeah, I remember seeing a paper published this year in Nature Geoscience. They also reached a conclusion that the AMOC wasn't fully collapsed or just slightly weakened during Heinrichstadial 1. And that's same conclusion in your talk. So it seems like this is a very robust discovery right now.
Yes, that was maybe Poppelmeier paper.
Yes, that's the one I was, yes.
Yes, and they also used a model with C13 in them, and I think other paleoproxies. Yes, so
yeah, I think they also use protactinium, thorium ratio, and some other proxies simulated model. Yeah, so yeah, it's time for question and answers. If the audience has any questions, again, please feel free to put them in the chat or raise your hand. And while people are still typing their questions, I wanted to ask I guess, simple question. So I remember, from watching Spencer's talk, he mentioned that for benthic carbon isotopes, you might need to do the correction for the biological effect. I was wondering for your comparison here, did you do that or?
No, well, the model so it's a process-based model of C13. So the model includes all the processes. It includes biological processes, biological fractionation. It includes fractionation during air-sea gas exchange. So all of these are included. So we compare then the model data to the observations. It's like an apple-to-apple comparison, what we're doing. But the observations were not corrected for any biological effects. No,
Okay.
but it's still, it's a fair comparison because we're modeling the proxy directly in the simulations.
I see.
And
Thanks.
yeah, well, these other factors, so the biology definitely affects C13, particularly in the LGM. I mentioned that quickly, I think, when I talked about the iron, but so if we do not add this extra iron in the Southern Ocean in the LGM simulations, we get too high deep ocean C13, and we also do not match the changes in the nitrogen isotopes. So both of these isotope systems are fit better in simulations that include iron fertilization. So that's the way how we constrain these biological effects through the nitrogen isotopes. I haven't showed our nitrogen isotopes here, but yeah, they are also in the model, and we have compared them with the paleo data.
Do we have any other questions for Andreas? Or if we have questions for Spencer, please also. Oh, John has a question.
I have a question for Andreas. Is there any hope to constrain the speed of AMOC?
Hope, there's always hope, right? Yeah, and I think, with maybe combining adding additional proxies. So what in our model so far, what we have explored is carbon isotopes. So we've used C13, also radiocarbon. I haven't showed any results for radiocarbon, but they're essentially very similar to the C13 results. So so the radiocarbon does not constrain strength either, but it constrains depth. But for the protactinium, thorium, and the neodymium, that remains to be seen how well these proxies constrain the AMOC. And we are just in the process of adding them to the model at this point. So we have not done any real LGM simulations or comparisons with data for those. But I think other groups have, including NCAR, right? I think you guys have the protactinium and neodymium in there. And I think the Bern group as well. But I don't know if anybody has really looked at how well these constrain the AMOC strength. But I think there is there's definitely hope, I would say.
Great. Yeah, if we have any other questions, again, please feel free to raise your hand or put in chat.
Another, regarding the previous question, another proxy that I think we should look into is the surface temperatures in terms of AMOC strength. I mean, the surface temperatures have, I think, the bipolar seesaw that is one of the I think the most convincing evidences that pointing to AMOC changes during the last glacial period. So maybe AMOC strength could be constrained with sea surface temperature data.
Can I ask another question?
Sure.
So yeah, I know that a couple of groups are trying to understand the impact of a weakening of AMOC during hydroxylated 1 on the hydroclimate part. And for some well, I think the data and model comparison reveals that we need to weaken the AMOC to a certain degree so that the model simulated hydroclimate change and proxy data are much better, such that we have a southward shift of ITCZ and in the monsoon region, where it's not like over changed or too weak a change. So do you think that can also provide some constraints there?
That's a good point. And I wonder if not only AMOC, but also like the effect of icebergs in the North Atlantic plays into that ITCZ shift. So I think, if you just cool the North Atlantic by putting icebergs there, even if you don't affect the AMOC very much, maybe that would be enough to shift the AMOC or to shift the ITCZ. So I think there, maybe you don't really need an AMOC collapse or a large reduction in the AMOC to shift the ITCZ. Maybe it's enough if you cover the North Atlantic with icebergs, cool it down, and that does the ITCZ shift. But yeah, I'm not sure, but it's definitely an interesting question.
Yeah, I was asking just mainly for discussion. And yes, thank you for providing your feedback. Yeah, do we have any other final questions? We already passed 11. So John, should we continue questions or should we end here?
If people can stay, I have a quick question for Spencer. So you mentioned the isopycnol mixing and dipycnol mixing, and those could be different for the last glacial maximum. I'm wondering whether we can use some other evidence to try to provide some hint about how those might actually change during the LGM.
Yeah, I think that's a really good question. And I think one thing that actually is not right. So at the moment, what happens in a fully coupled climate model is usually you choose some values for isopycnal mixing. It used to be a lot, maybe 10 years ago that people would choose the same value everywhere. Now they choose, they know a bit more about this, right? Somebody has gone out and done high resolution modeling and thought and made observations and there tends to be higher isopycnal mixing set near the surface and higher isopycnal mixing is set in places with stronger winds. But usually it's not a very dynamic parameterization. So if the winds change, right, the actual amount of isopycnal mixing you get in the ocean, it does not really respond. And so we can start to move towards a place where we have a more dynamic understanding of what sets the isopycnal mixing, right, and that will help us to have a parameterization that's perhaps a bit more responsive to these kinds of things that might have changed at the LGM. But still, right, we would need to know, like, are we modeling the winds well? And that is still a thing, right? If you go and look at different models, they have different winds at the LGM. So yeah, I think I think probably I agree that all is not lost, but I also often look at these problems and think, oh my goodness, there's just so many processes, and like even in the modern day, we have we have to have so many different groups modeling using different models and checking that they all agree in order to feel certain that we're doing the right thing. And one really big challenge that we have here is that like there just aren't that many people doing this kind of modeling, and so it's hard to feel certain that we are covering all of the parameters that there are. I mean, I would actually quite like to hear what Andreas has to say about that, but yeah, that's yeah.
So Andreas, do you want to add your comment or?
About what exactly?
I mean, I was just about feeling like there are so many, we need so many different models in order to be certain in the modern era. So do we need that? Do we need to be doing that diversity of modeling at the in paleoclimate? And is that even possible? How do we navigate this? I know that's quite a big question.
Well, I mean, in general, having different models is useful to get an idea about the uncertainty that is in the models. But then also, not every model is the same. So that some models may have some processes that other models don't. And in particular, I mean, these days, and if we talk about the last glacial maximum and things like that, none of the models have interactive ice sheets, I think. I mean, maybe there are some, but so there's still some components of the climate system that are not interactive. And that is, I think, a big issue too. So yeah, I guess that's all that I have to say to this.
Hey, Sophia?
I have one, I guess maybe this will be the final question. And I'll try, this question is a bit kind of like, give you both the floor to kind of, I guess, wrap up this kind of discussion, which is, do either of you feel that there is a proxy or a modeling capability that is needed to kind of reduce this parameter space? Like perhaps the parameter space hasn't been fully explored and perhaps there's very high uncertainty in AMOC magnitude. Is there anything we need to reduce that uncertainty that you feel is lacking right now in either modeling capabilities or proxy development? It's kind of a very broad question, but just general thoughts on that. I'm curious to hear your opinions.
Spencer, you want to go first?
Sure. I mean, I think, yeah, like I said in the talk, right, I am the thing that I'm excited about is the possibility that we can really cover the whole of parameter space, or a lot more of parameter space, using some more analytical, simple models, and then pick, right, oh, we think maybe this kind of area of parameter space is interesting and it seems like it might fit the data, and then you can go and run a more three-dimensional model with more processes in that kind of area of parameter space. And so that's something I think is a good idea. But I mean, the truth is, I think we need people to do a really wide variety of things. And I also can't talk very much about proxies because I'm not an expert on all of the proxies out there. But yeah. Yeah, I think analytical models are a good tool that is underutilized.
And I'm not a proxy person either, so I don't have any ideas what to do on the proxy side. But on the modeling side, I think to me, it is very clear what we need to do. We need to add more of those paleo data, tracer data in the model. So for example, in the PAWS UVic model that I'm working with, we still don't have O18 in there. So, and there's lots of O18 data, right? And O18 is also coupled with the whole hydrological cycle, including the ice sheets, right? So you need it in the ice sheet model, in the ocean, in the atmosphere. And then that would provide, I'm pretty sure that would provide some additional constraints, how much, that remains to be seen. But adding all these different isotopic and other tracers and trying to find like a model scenario, a state or a changing state that fits all of these data simultaneously, including the ice core data, that is really a challenge for the modelers, because at this point, there is not a model that has all of that in them, including the interactive ice sheets. So there's a lot of model development to do.
In the interest of time, we should conclude here. Let's thank the speakers again, and thank you everyone for staying the extra minutes here. Thank you. So stay tuned for the next webinar. Bye.
Thank you.